which is going to be focused on women in science. Very excited about this session in particular. I want to just give you a few things for technical, in case you have technical difficulties, please contact Shilpa Narina, who's the session tech. You can contact her through the chat, but please do not use the Q&A. The Q&A will be for our panel questions or our speaker questions. At the end of this session, we're going to have a poll. And so I encourage you to, at the very end of the session, we're going to have a poll. So I encourage you to answer that. Additionally, we're going to have, so the questions will come through the Q&A section. And then also, please try not to leave the session if you don't have to, because it's disruptive to the speaker. So please try to stay for the whole session if you are at all possible. So with that, I'm going to go ahead and start the session. I'd like to introduce our first speaker. It's going to be Mary Ann Liebert. And she's going to tell us a little bit about the Rosalind Franklin Society and also the namesake of the Rosalind Franklin Award. Mary Ann Liebert is the president, CEO, and namesake of the privately held publishing company, Mary Ann Liebert Incorporated. She's widely recognized as a visionary in the professional publishing industry, and she has received many awards, too many to even name here. One of the things I'm super impressed by her is that she is the founder of the Rosalind Franklin Society, which recognizes, fosters, and advances the important contributions made by women in life sciences and related fields. So now I'm going to ask her to unmute herself, and she is going to tell us a little bit about the Rosalind Franklin Society and Rosalind Franklin in general. So I'm going to welcome Mary Ann to the virtual podium. And you're muted. Sorry. Go ahead, Mary Ann. Go ahead and unmute yourself. Oh, sorry.
Okay. I'm really delighted to be here, and the Rosalind Franklin Society has been such a big imperative for me. And the thing that I like the most is that we are making a difference. Why did we start? Well, first of all, I have scientists in my family, but primarily in the physical sciences, although my interest has always been in the life sciences. So I always followed the life sciences carefully, and we had started a publication called Genetic Engineering and Biotechnology News. That was my first publication. And when Nancy Hopkins had taken on Larry Summers, as everybody knows, when he felt that there was an intrinsic difference between men and women in the sciences and the mathematics, I was equally incensed. Nancy said she was either going to walk out or throw up, and I said, I've got to do something. So I'm going to do it now. I'm going to start a society, and I named it after Rosalind Franklin because I felt that with the double helix, she had been really deprived of the recognition that she should have had. So what was our mission, and what is our mission? Our mission is to ensure more tenure-track positions for women and more nominations and awards for women, so that women have not only equal opportunity, but equal recognition. Now, I must say that once I started, when we had our first meeting, by the way, the two things that came up as the most important things were childcare and having positions on corporate boards. That took so much of the meeting. And the late Mathilde Krim used to say, the cell waits for no one. And that's what a lot of people felt. And so I was determined to help move this forward and do everything we could. So we funded it. And with the help of our executive director, Carla Rubinger, we were able to make things happen. What has happened? We advocate very strongly when we know there is a group that doesn't have a woman on the judge panel, we make a call. If women are not considered for prizes, we make a call. And by the way, we have 5 Nobel laureates on our board. I'm really proud of that. And for the founding board, I asked everybody personally to be on that board. Now, interestingly enough, Nancy declined. Here she was, my impetus for starting, and she had declined, but she came on later. I will say, by the way, that as a biomedical science publisher who's so interested in this field, we have over 90 journals, and only about 15 of them are edited by women. That's been the case for all these years. We try, but for whatever reason, it is harder to get women. We're trying now to focus on more diversity. Our board is not diverse. We have made that a priority for something now. Our meetings were scheduled in the very beginning to be very small and very intimate, to give women a real chance to express their real feelings, how they felt, what worried them, and to speak openly. The meetings have grown. This year it was supposed to be at the Howard Hughes Medical Institute, but because of COVID, that's not possible. But I will say, and I'm proud of this, on Genetic Engineering News, if you can see this cover, it's all about COVID-19 research. Women are changing the face of the pandemic, and we are determined to keep women in the forefront. When I started Genetic Engineering and Biotechnology News and said to the editor, let's feature some women in biotechnology, it didn't happen. But we do now, and so having women advance in biotechnology is a major imperative of Genetic Engineering and Biotechnology News. And we would love more women to be editors-in-chiefs of important journals. We have DNA and Cell Biology that's edited by a woman. We have Phage that is edited by a woman, but we need more. And we are determined to help women move up the needle. I will say, by the way, that men, and the Rosalind Franklin Society is not just for women. Men are on the board too, which sets us apart from other organizations that we're all women for women, and we are not. And Torsten Wiesel was one of the first to join, and Tom Check. So we've had, interestingly, and Linda Buck is on our board. And when we asked her to present on what it was like to get the Nobel Prize, she said, nobody's really interested, and she didn't come. So Richard Axel came, and he told us a terrific story that was entertaining and informative. And we want to see more women be nominated for major prizes, and we want to see women get the best tenure track positions they can do. Women need to advocate more for women. They are not good advocates for women. Some are, but most aren't. Maybe it's because the top of the needle is so thin, and because they are still the ones who are whose major responsibility, the health of their children and their child care falls under. But we are making a difference. We make calls, we have a newsletter, and there's nothing quiet about us. So I want more women editors of our peer-reviewed journals. We want to make a difference in every way that we can. We have a newsletter, and we're celebrating Rosalind Franklin's birthday because she deserves to be celebrated. In this issue of Genetic Engineering and Biotechnology News, Kevin Davies, Dr. Kevin Davies, has written an extraordinary piece about her. And when she was alive, by the way, Brenda Maddox, who wrote the biography of Rosalind Franklin, flew over to our meeting and spoke to our group. And Francis Collins has had dinner with us and played his guitar, Rosalind, for us when our group was smaller and we used to have our meetings at the Pew Foundation. So now we have to decide, how much do we grow? If we grow too much, we lose the intimacy and the ability for the members to really speak to one another. But as the membership has aged, they don't seem to need it as much. So we have to look for the next generation of activists to work with us and help keep Rosalind Franklin's name right in front 100 years and beyond. Thank you.
Thank you so much, Marianne. And now we're going to move over to Erin Nolan. She's going to introduce the award, what we call the Rosalind Franklin. This is the inaugural year of the Rosalind Franklin Award. And she's going to introduce the finalists.
I'm here to introduce the Rosalind Franklin Society Medal, the Genome Writers Guild, and Rosalind Franklin come together to create the Rosalind Franklin Medal. This award marries together the Genome Writers Guild core objectives of facilitating genome writers' conversation, collaboration, and exposure with Rosalind Franklin Society's goals of enabling more women to achieve higher recognition, visibility, appointments, and success in industry, academia, or government. The recipients of this inaugural award embodies the missions of both groups. First up, we have Jessica Cortez as a nominee. Jessica Cortez is from the University of California, San Francisco. Next, we have Fatima Anam. She's from Stanford University. Next, we have Natalia Gomez Aspina. She is an MD PhD from Stanford University. Next, we have Jennifer Hamilton from the University of California, Berkeley. And finally, we have Alexis Komor from the University of California, San Diego.
All right, I wish I could have a drum roll here. We have the award, so Aaron can hold up the actual award. We don't have it to hand off to the winner, but we do have it. It's very lovely. You can see it here. And the winner is Dr. Alexis Komor. Yay, I'm cheering for you. I'm sure everybody is cheering for you as they come up. So I'll introduce Alexis and some of the things that she has done. Dr. Komor has completed her postdoctoral work in 2004 in the laboratory of Jacqueline K. Barton at the California Institute of Technology, where she worked as an NSF graduate research fellow on the design, synthesis, and study of DNA mismatching binding metal complexes. She pursued her postdoctoral work as the Ruth L. Kirstein NIH postdoctoral fellow in the laboratory of David Liu, where she developed, as everybody probably on this call knows, base editing, which is a technology that allows us to make point mutations without using double-stranded breaks. Alexis joined the Department of Chemistry and Biochemistry at the University of California just in 2017, where her lab develops and applies new precision genome editing technologies to the functional genomics field. I don't think I really had to introduce Alexis to this group because she has made such an impact to genome editing, especially over the last 3-4 years. One of the things that really made Alexis, Dr. Komor, stand out was that she has done not only all of these great scientific achievements, but she's also done a lot of things for outreach, especially for women in science. So Dr. Komor orchestrated an outreach program with the San Diego Better Education for Women in Science and Engineering community called Be Wise, where over 50 middle school aged girls came on campus and they toured her lab and the labs of 4 other female faculty members. Additionally, while she's been setting up her independent lab, she has also developed a genome editing outreach laboratory activity for the local San Diego high school students. Dr. Komor was also nominated by 4 individuals that were prominent individuals in the genome engineering field, one of whom was Dr. David Liu, who was her mentor for her postdoctoral research. And I thought one of the things that really stood out to me, besides all of her accomplishments, besides all of her outreach, was the fact that, you know, David Liu, who is a genome engineering god in his own respect, he called her a coworker. He didn't call her a trainee, he called her a coworker. And not only did he call her a coworker, he said that she was one of the best coworkers he's ever had. So with that, we're very happy and honored to have her here. Congratulations. And I'm going to turn over the screen to her, and she's going to present a talk about her recent research. Alexis.
Very beyond honored to be here, and I hope I can do her justice with this talk. So I want to talk to you guys today about the work that we've been doing in my own lab over the past 3 years. Oh, there we go. And it's in the functional genomics field. And so what that means is that we strive to understand the functional consequences on cellular function and human health of human genetic variation. And so, as you all know, everyone has DNA, and it is what makes us unique. If we compare the genomic sequences of 2 individuals, we'll find that they differ from each other at a lot of places. If the sequences differ just by the identity of a single base pair, for example, this person 1 has a C and person 2 has a T, this is called a single nucleotide variant. And single nucleotide variants are responsible for about 96% of human genetic variation that's observed through human genome sequencing studies. And so what's more, as I mentioned, these genetic variations make us unique. They can also have a negative impact on our health. If we look at the sequences of a reasonably genetically or ethnically diverse group of people and compare them, our typical genome will differ from the human reference genome at around 4 to 5 million sites out of 6.4 billion base pairs in our genomes. And so it's tempting to think about this in a very black and white sense. So, okay, this single nucleotide variant here is benign, it's just contributing to diversity, versus this red one here, that's pathogenic, that's bad, that causes disease. But the reality of the situation is that it's more in shades of gray. So when we classify these variants in terms of their impact on our health, they kind of fall into these 5 different categories: benign, likely benign, variant of uncertain significance, or VUS, likely pathogenic, pathogenic. And what's more, if we look at all the different genetic variants that we have identified so far, less than 1% of them are actually given a classification, okay? And this is known as the variant interpretation problem, okay? If we look through all of this genomic or human genomic sequencing data, 99% of it is unclassified. We have no idea what the functional consequences of these variants are. And then of this less than 1% that we can give a clinical classification to, you'll notice here, it's a very small percentage where we say this is definitely benign or this is definitely pathogenic. And this is because the way that we currently can give these variants classification is by sequencing lots and lots and lots of individuals, and then saying, oh, well, this variant has popped up in enough diseased individuals that we can say it's pathogenic, or it's been observed in so many people that are healthy that it's benign. And this is problematic because if we look at the ethnicity of the people who are being sequenced and who we are sort of classifying these variants based off of, nearly 90% of them are white Europeans, okay? And so these sort of traditionally marginalized populations are also grossly underrepresented in these studies. And so this is sort of the basis of precision medicine, is these classification systems where a person will come to the clinic, they're sick, we sequence them and we say, oh, we found a pathogenic variant and we know what it's doing, so we know how to treat this person. The majority of those variants where we know that they're causing a disease and we have an idea of how to treat them are specific to these European populations. So there's a huge issue with the current system of how we classify these variants. And so what we strive to do is to develop methods where we can study them in a laboratory setting, okay? And then all of these variants are sort of created equal in our mind. It doesn't matter what type of variant it is, we can introduce it into a cell line and then look at how that is impacting cellular function. Okay. And so the way that we would do this, we have to have the tools to introduce the variation into the cells in the first place. And so given this is a Genome Writers Guild conference, I probably don't need to tell you about this slide in great detail, but traditional genome editing methods to do this would rely on programmable nucleases, and we introduce a double-stranded break. And then the bottom line is that these double-stranded breaks are very toxic. Indels will dominate the pool, and so if you're trying to do precision DNA modification, it's really problematic, and especially because this HDR pathway is very cell cycle dependent. So depending on what cells you're working in, it can be even more difficult to get your specific single nucleotide variant introduced into your cell line. Okay, so that brings me to the current lab research. The bottom line is sort of, we want to understand the functional consequences of these single nucleotide variants. We want to understand how they're impacting human health, potentially understand and generate better treatments or cures for these. And so this is one subgroup of the lab, but so what we do is we look at, oh, here's a particular variant we want to understand. We need to be able to generate, we need to have the tools to introduce the variant. So another subgroup is we need to engineer these precision genome editing tools to sort of allow this functional genomics subgroup to happen. So those kind of feed with each other very well. Another thing is that a lot of these, I call them non-traditional genome editing tools, they're very understudied in terms of the mechanisms by which they work. And so when we look at these tools, we want to better understand that. So we have another subgroup looking at how are these tools working, both from enzymatic perspectives in terms of, you know, how is Cas9 binding, or how is this enzyme actually modifying the nucleobases, as well as from a DNA repair standpoint. Once we introduce some sort of modification into the genome, what is the cell doing? And so in that way, it sort of feeds back into this engineering subgroup where we can further optimize or engineer new tools based off of what we just understood. And finally, just because we play to our strengths, we're DNA repair people, some of the functional genomic studies that we are doing right now are looking at DNA repair proteins. And so sometimes these two subgroups will actually synergize with each other as well, just because of what we're understanding about DNA repair through these mutational functional studies. And so at the crux of these is obviously base editing, which I'm sure you guys have heard about. And so in this, I was sort of thrown into the genome editing field as a postdoc and didn't really know what I was getting into, but I was a nucleic acid chemist, and I knew that if you want to make a small perturbation to the sequence of the genome, it didn't make sense to me to cut the DNA backbone. And so very early on, we said, okay, what if we take Cas9? It's very easily reprogrammed. And instead of allowing it to cut the DNA, we inactivate it so it binds but doesn't cleave. And then this is the crux of everything, is finding these enzymes that can modify nucleobases in DNA in some way that will overall catalyze a point mutation introduction. And so, like a good chemist, I stared at the crystal structure of Cas9 bound to its R loop for a really long time. And we realized that we could take advantage of this sort of single-stranded portion of the R loop. And so, ever since then, I've been completely obsessed with finding these nucleic acid modifying enzymes that are specific to single-stranded DNA only. And the generation of the first base editor was relatively straightforward because there was a class of naturally occurring enzymes that could do this, cytidine deaminases. And so this was CBE, the first double-stranded break-free genome editing agent. And so we have a Cas9 nickase, guide RNA, so we can program it to do genome editing wherever we want, and then a single-stranded DNA-specific cytidine deaminase enzyme. And so we bind to DNA, we open it, and we catalyze C to U conversion down here, and we nick this top strand here to try to trick the DNA repair pathways in the cell to use this bottom strand as a template when fixing this lesion. Okay, so uracil has the same pairing properties of thymine. So this was CBE. It can take a CG base pair to a TA base pair with very high efficiencies and precision. And before we had even gotten the story out, we realized that adenosine was amenable to the same type of chemistry. So we can deaminate adenosine to inosine, and inosine is read as guanine by polymerases. So in theory, if we had, again, a single-stranded DNA-specific adenosine deaminase enzyme, we could deaminate adenosines to inosine to give an overall AT to GC base pair conversion, which is sort of the complement of what we did before with the CBE. The issue was there were no more naturally occurring enzymes that were specific to single-stranded DNA that could alter the base pairing properties of a nucleobase. But there were some of these enzymes that existed that could do this chemistry in RNA. And so we embarked on a really crazy directed evolution project where we started with this enzyme. It's called TAD-A. It's from E. coli. And this is the crystal structure of it. It catalyzed this adenosine deamination chemistry in the loop region of a tRNA. Okay. And if you look here, it binds as a dimer. This subunit here in dark gray is doing the chemistry, and this lighter gray subunit is just helping with binding. And all of the contacts between the protein and the nucleic acid substrate were confined to the loop region. So no interactions with the crazy cloverleaf structure or the stem portion. So it was like this enzyme was a single-stranded RNA modifying enzyme. We thought that was close enough. So we would mutagenize the TAD-A gene, ligate it into this backbone where it's fused to dead Cas9 and then given a guide RNA so that when we transform it into these selection E. coli, they have a broken antibiotic resistance gene in it, and it's due to an inactivating G to A point mutation. And so the guide RNA brings our library member to this protospacer, and any functional library members that are able to do deamination chemistry on single-stranded DNA could take this A, which is right in that sweet spot of the protospacer, to a G, and then we plate on varying concentrations of antibiotic, sequence the surviving colonies, and find mutations in TAD-A that allowed it to do this chemistry on DNA, because the wild type enzyme had no editing activity at all on DNA. And so that's where I'm going to start on now with what my current lab has been doing. So this is TAD-A. Over 7 rounds of directed evolution, we identified 15 beneficial mutations. And what we think about now is that, okay, if we want to engineer new types of base editors or new types of genome editing agents, we need to learn from this story, because we ran out of naturally occurring DNA editing enzymes. If we want to make more, we have to engineer them. So again, CBE on the top, ABE on the bottom. Another thing I want to point out, which I don't have time to talk about today, but I purposely have this arrow here with all these question marks above it. We have no idea how this GU or TI intermediate is processed by the cell. Okay, how does it go from this TI intermediate to a CG base pair? And so that is one of the focuses of our lab. But today I want to talk about this TAD-A, and I want to talk about what the heck these different mutations are doing. As I mentioned, wild type TAD-A has no activity at all on DNA. When I started my lab, there's this very, very talented graduate student, Kartik, who's technically in Francesco Pisani's lab, who is a computational chemist in my department. And she said, I want to do molecular dynamic simulations and figure out what's going on. How do we turn this enzyme from an RNA editing enzyme to a DNA editing enzyme? Can we kind of harness that information to guide future studies into engineering future nucleic acid modifying enzymes? So the mutations that we picked up during the evolution are shown here. Round 1 are shown in red, and then as we go up to higher and higher rounds of evolution, those go to purple. And in particular, we focused on these first round mutations, specifically this aspartic acid to asparagine mutation, because that was sort of this linchpin in the directed evolution. When you start a directed evolution project, you really, really, really want to start with non-zero activity. If you have 0 activity, it's really hard to get the project to work. And we started with 0 activity, but this mutation here gave us non-zero activity and allowed the project to actually be successful. So what is it about this mutation that allowed the enzyme to be able to do its chemistry on DNA? And so Kartik did a couple of different types of molecular dynamic simulations, both steered and not unbiased. Here, she just took the protein, did 500 nanoseconds of unbiased simulations to look at what the overall structure of the protein was. And so shown over here is the root mean square fluctuation of the different amino acids within the protein. So it's basically telling us over these 500 nanoseconds, how much are these amino acids moving around? And there was this really important loop region, so you can see it right here in the movie, between the beta 4 and beta 5 strands, and it had really high fluctionality. So it was moving around a lot in the wild type enzyme. When we installed the first mutation, again, this D108N mutation, there was a drastic decrease in the root mean square fluctuation. And in fact, this loop started to gain some helix properties. Okay. If we ran the same simulations, again on wild type TAT-A, but previously this is all the monomers. Remember I said that TAT-A does its chemistry on its RNA substrate as a dimer. If we dimerize 2 wild type TAT-As, we see the same decrease in fluctionality of this loop region here. And so we said it was maybe this mutation is sort of tricking the enzyme into thinking it's dimerized. And so we said, okay, here's the ABE 7.10 construct. Okay, we have a wild type TAT-A fused to a mutated TAT-A that has all 14 mutations in it, fused to Cas9 nickase. If we instead have the mutated TAT-A here, there's actually a huge decrease in editing activity. And we think it's because this was sort of that binding monomer. It wasn't involved in catalysis. And if we had all of these mutations in it that were responsible for this increased catalysis on DNA, they weren't really necessary, and it just destabilized the protein. So the final construct that we had engineered originally was wild type TAT-A, mutant TAT-A. And so we truncated off the wild type TAT-A to give just the mutated TAT-A monomer fused to Cas9. And we found that they had basically identical editing activity across a variety of different sites. And so we said, okay, we're going to take this to mean that we have mutated this enzyme so much that we basically changed its sort of fundamentally how it's doing its catalysis. It's doing its DNA editing as a monomer. This was also great because we then wanted to do simulations to look at how TAT-A is interacting with the DNA substrate, and the entire ABE complex with the DNA and the Cas9, everything was way too big to do simulations. So we just minimized the system to a TAT-A monomer and a piece of DNA where we put constraints on the DNA so that it has the same overall structure of the R loop. So we're keeping the 2 ends of the DNA at a particular distance apart from each other. And then we do a couple different simulations. And here, these are steered simulations where we take the DNA and we push it into the active site of the protein, and then we pull it away and we see how difficult it is. So how much does the protein want to hold on to that DNA substrate? Okay. So this is showing the distance between the center of mass of the protein and the nucleic acid substrate. And what is important to note is that as we start pulling the DNA away over here, this sort of free energy, the energy required to do that is not much. This has a very weak slope here. And if we just install that first round mutation, the D108N, we see about 20 kilocalorie per mole increase in this in the energy required to pull it away. So we've increased the binding affinity substantially. And if we then start putting in these additional rounds of mutations that we identified in the directed evolution, we actually don't see an additional increase in the binding affinity. And so when it's a base editor, Cas9 is helping it to bind to its nucleic acid substrate. So it doesn't need a really great binding affinity, but it does need something that's better than the wild type TAT-A. Okay. And so if we do unbiased simulations where we just let the protein interact with the nucleic acid substrate and then look at what's happening with the individual amino acids, over here, this is wild type TAT-A. We're trying to force it to interact with DNA essentially. And what happens is this aspartic acid residue, when this is RNA, there's a 2' hydroxyl that this aspartic acid can hydrogen bond with. And when we take the 2' hydroxyl away, because now this is a DNA, there's nothing to sort of quench that negative charge. And so it rotates back towards the active site zinc into a very energetically unfavorable conformation to get away from that negatively charged backbone. And when this is mutated to asparagine, the negative charge is gone, so it rotates back over, and now it's in a more energetically favorable conformation, allowing for DNA binding. And as additional rounds of mutations are incorporated into the protein, we get these sort of allosteric effects leading to local rearrangements around here. And this eventually does make a hydrogen bond in contact with the DNA backbone. So Kartik wanted to say, okay, how important is this D108N mutation? So she ran these simulations again with the higher generation ABEs. So these had additional mutations that had been picked up during first and second round of evolution. And she reverted that the N108 mutation back to D, so from asparagine to aspartic acid again. And these are shown in green and blue here. You can see, even though they have 1 or 2 or 3 beneficial mutations in them, just the loss of that one asparagine 108 mutation is enough to make them have binding affinity that is similar to wild type. And so when we went into the lab to test this, we generated these base editor constructs and then looked at what their base editing efficiency was at a couple of different sites. And so you can see the first generation ABEs in red and then second generation in orange are here. And then when we just mutate this N108 back to D, they have wild type like activity. So we see almost complete abolishment of the editing activity, which suggests that Kartik's simulations in this platform are actually really emulating what's happening in real life. And so one other thing we wanted to look at, we said, how important is it? What about in the AB710 background? So in these 2 pinks, these are the AB710, the dimer and the monomer, and then in turquoise and in yellow, what we've done is we've reverted that N108 back to D, but there are still 13 different beneficial mutations in there, and yet the editing activity is almost completely abolished. And so, as Kartik likes to explain, not all mutations are created equally. As we have gone through this directed evolution, we've sort of built up and identified all these mutations and led to an editing agent that has nearly 80% efficiency at certain sites. And if we just take out one of these mutations from the earliest round, it all just falls apart, and we're left with a dead enzyme. And so this was really shocking to me. It made me realize how lucky we were that that library that we generated in the first round of our evolution had that mutation in it. Okay, and so this really highlights how with these directed evolution campaigns, having that non-zero amount of activity to begin with is so important. So I want to kind of finish up some of the research side of the lab. I want to make sure I have time to tell you about the outreach that we've been doing. But I want to touch upon one of our functional genomics studies that's been done by Carlos Vasquez, who's a Ford Fellow in the lab. And what he's looking at is DNA repair protein that's involved in oxidative DNA damage repair. So GC base pair looks like this. When the cell is exposed to reactive oxygen species, guanine is always oxidized. It's the most easily oxidized base. It can still base pair with C like this, but if if translation or transcription occurs over it, or replication, what happens is the 8-oxo-G will flip into a Hoogsteen base pair, and it will show this base of the base over to its partner, and then adenine will get misincorporated across from it. And so overall, this can lead to GC to TA base pair conversions. There are two enzymes that are responsible for preventing these mutations from accumulating in our genome. One is MUT-YH, which will recognize 8-oxo-G when it's mispaired with A. It will excise the A, and then a C gets put in, and then OG1 recognizes the 8-oxo-G when it's mispaired with a C, and it will excise the 8-oxo-G. And so just to sort of highlight, again, this variant interpretation problem, if you look at mutations that have been identified in MUT-YH, almost all of them are variants of uncertain significance. So we don't know exactly what they are doing in terms of human health, but they are associated with disease. And in fact, mutations in this gene are associated with MUT-YH-associated polyposis, or MAP, which is a colorectal cancer. And it's important to note that people who have this disease, they're always homozygous in terms of their mutations. Okay. And so what Carlos is doing, and so we've written up this protocol draft, and it's currently under review, is we've realized that generating isogenic cell lines, which is when you generate a cell line where, compared to a parental line, it only differs by the presence of a single genomic modification of interest. Okay, it's a really, really powerful tool for functional genomic studies, because you can tease apart what the consequence of just one particular type of mutation is. And so what they do is this is headed by Carlos, and also Quinn Cowan helped with this. We will design a particular guide RNA base editor combo. We'll transfect them into cells and then validate that they are installing the mutation that we want. So this is through just using Sanger sequencing on bulk cells. We can see editing that's around 10 to 20% or so. We're trying to mutate this T to a C using ABE by editing the A on the other strand. The base editor is transcribed on the same mRNA as a GFP. So what we can do is then take those cells, bring them to the flow cytometry core, and use FACS to isolate a single GFP positive cell per well in a 96 well plate. And then you allow the cell to generate a colony, and that is your new cell line. All of those cells originated from the same original cell, and if you're lucky, they will have your point mutation of interest. And so typically for a 96 well plate, it will yield about 8 clones. Okay, so the cells are not super happy with the situation that we're doing to them. But what we found is that about 50% of the clones end up being wild type or unedited. Especially if we have about this editing efficiency to begin with, we'll get 25% to be heterozygous, and then 25% to be homozygous. And in direct contrast, if we were trying to use HDR to install point mutations specifically, you generally have to sequence hundreds of clones, and you'll get a heterozygous mutation if you're really lucky. And so, as I mentioned, this particular disease, MAP, is only caused by homozygous mutations. So we wouldn't even really be able to model that disease state if we weren't able to generate these homozygous cell lines. And so, quickly to wrap this up, these cell lines also recapitulate the reduced MUT-YH activity that we would expect. And the way that we assess this is that we generate this plasmid where we have mCherry, again transcribed in the same mRNA as a GFP through a self-cleaving linker. So they're transcribed in the same mRNA, and then they translate to different proteins. And we use sort of combination of chemical biology tools and molecular biology tools to install an 8-oxo-GA lesion specifically into codon 35, such that transcription across this template strand down here results in a premature stop in GFP. Okay. So we transfect them into cells and then use flow cytometry to look at mCherry to GFP fluorescence. And so these are positive and negative controls. If the ADOXO-GA lesion is not repaired, so we have an A in the template strand, you will only see mCherry fluorescence. That's up here. And if the cell is able to repair this bottom strand back to a C, okay, and so again, that's MUT-YH's job, is to excise this A and initiate the incorporation of C there. Now here you can see that a large percentage of the cells have both mCherry and GFP fluorescence. Okay, so if we take this reporter and transfect it into wild type cells, we can see 83% of the cells are repairing the ADOXO-GA lesion. If we look at our heterozygous cell line, it has similar activity to wild type, about 76%. And if we look at the homozygous cell line, though, we see a large decrease in editing activity, or in ADOXO-G repair activity compared to wild type. Okay. And this is a known pathogenic mutant. We've looked at several different VUSs, and we're able to make what we think are pretty educated guesses as to what the clinical interpretation should be. And so we're really, really excited about this and sort of building out this subgroup of the lab. All right, so in the last 10 minutes, I wanted to touch upon our outreach efforts. There are few scientific disciplines, such as CRISPR, that have, really taken non-scientists by storm and have, sort of been bombarded across the news. And so we think that the genome editing is a really, really good way to get young people excited about science. And so we've initiated several different outreach programs that are all sort of focused at the pre-college phase, when we think that, it's not too late yet. And so the first one was through the Better Education for Women in Science and Engineering chapter in San Diego. This is a picture of us where 50 middle school aged girls came to campus, and we gave them tours through labs and hands-on activities. We took the opportunity to sort of teach them about genome editing. So we had some bacteria that were expressing GFP, and we showed them how just by a single point mutation that can be installed with ABE or reverted with CBE, we could change GFP to BFP. And then we took some of our culture plates and decorated them with fluorescent bacteria to show them. It was a really fun day. It was really awesome. And then we sort of thought, okay, how do we make this bigger? How do we sort of build this out? And so that's when we developed this 3-day lesson plan that is aimed at the local public high schools that have really high numbers of underrepresented minority students at them. And what we do is we teach them about genome editing, and then we bring in these E. coli cells where they have what we call GFP-itis. Okay, so they have GFP, but there's a point mutation in it that makes it non-fluorescent. So the fluorophore here has been mutated, so they're not fluorescent. And we kind of tell them, oh yeah, it's similar to like sickle cell anemia. Okay. And then we give them a choice of 3 different guide RNAs to choose from. So they have to have paid attention during the lesson plan and pick the correct guide RNA to transform into these E. coli to try to fix their GFP-itis. And if they do, then we get an A to G mutation down here, and then the cells glow again. And again, you can just visualize this with a handheld black light. And so we're super excited that this is what it looks like. Here's the sort of negative control with no base editing at all. If the students pick the wrong guide RNA, this is what the cells look like. And then if they paid attention and they picked the right guide RNA, this is what the plate will look like. And so we think this is really, really cool. And it should get students really excited about science who may not have been excited about science to begin with. And so with that, I want to have a shout out to my group They're awesome group of individuals. They're shown here. I talked about Kardic's work with the computational stuff, and then Carlos is working on the MUTYH project. They're all involved in our outreach efforts. It's wonderful to get to see them, although I haven't been able to actually see them lately just via Zoom. But before COVID happened, I couldn't ask for a better group of people to work with every day. So I want to thank them. Here is our Twitter handle, so please come follow us on Twitter, and then these funding sources. Again, thank you so much for this award. It is such a great honor, and I hope I did Rosalind Franklin proud.
I think you definitely did. And thank you for that great talk. And we will go ahead and take some questions from the audience. So I just want to remind you, if you have questions for Alexis, to go ahead and add into the Q&A. I'll start us off. It's an easy one. I'm sure you get to ask this a lot, but with base editors, there's this bystander effect where if you have a base pair, an A, let's say, next to it, you're going to get a bystander effect. Is your lab working on that? And are you making any progress?
We're thinking about it. I honestly think the best way to combat that is to just have a sort of a slew of different base editors where each one has a different sequence motif context. So, you have a CBE that goes after TCs, another one that goes after GCs, et cetera, et cetera. And then same with the ABEs. I think that is honestly the best way to go. With a lot of there's so many of these genome editing tools now that we've kind of gone, I think the field as a whole has realized there isn't one size fits all type thing. And particularly if you're thinking about therapeutics wise, it's best to just, find the absolute, best one for the job and go with that. So I think that's sort of where we're headed.
Great. And then we have another question from Scott McIver that says, are we going to be able to make transversions?
Hopefully, we're working on it. So there's two ways to think about it. One is by, and so there are a few papers that came out earlier this week where you use an enzyme that we already have, and then you try to manipulate DNA repair to give you different outcome based off of that GU or that IT intermediate. And so that's one of the things in our subgroup that's looking at these DNA repair mechanisms of base editors is, can we identify what the genes are that are involved in these different outcomes? And then another thing is to open up the chemistry. So as I kind of showed you, this is funny how all the subgroups kind of work together. You can have certain types of modified bases that will flip into a Hoogsteen base pair conformation, and that allows for transversions. And so it's about identifying the right type of chemistry and then the corresponding enzyme. And so we're hoping that these studies on TAT-A to tell us, okay, molecularly, what do we need to do to flip an enzyme from an RNA modifying enzyme to a DNA modifying enzyme? Because there are tons of RNA modifications that are installed by different enzymes. So I think it's those, two different pathways to go down.
Okay, we have another question from Lynn Fellman, who she wants to ask, does your research interest in human genetic variation intersect with the NHGRI's efforts to build a pan-reference genome? Are you working with them?
We're not, no, but we're trying. We would like to get some money from them, yeah.
Yeah. All right, and then a question specific to your being a woman in science. What is the biggest problem issue you have encountered as a woman in science?
Me personally is harassment in the workplace, and it's happened time and time again. Doesn't multiple different places where I've been, where it gets to the point where it's gotten to the point where I didn't feel comfortable going to work, and I know that most of my male colleagues have never felt that before. And I know there are lots of different things that other people have experienced. That's my personal experience has been that has been the toughest thing to overcome to get where I am today.
Thanks for sharing that. And we'll have more questions for you, and we're going to have a panel with the rest of the panelists in a few minutes, but we have a couple more science questions. or questions regarding your talk. What would you recommend as a first step to start a science outreach program for school students and the general public and the community?
It's hard. So we sort of just, we bit the bullet and we used my startup fund because it's expensive to make these constructs. The schools that really need the outreach don't have the money to be able to have the lab experiments. But once you get some of this preliminary data, then, and UCSD has been really great too. When I sort of shared with them what our overall goal was, they said, oh yeah, we definitely have money for that. Here, use this. But they're, NSF, broader impacts, they take this really seriously. But it's kind of hard to find the outlet sometimes too, but I definitely think that the public schools are definitely ones that they need it.
Well, thank you so much. And again, I want Erin to show we've had requests to show the award again, which we will mail to you. I don't know if Erin can show the award.
Can I just say congratulations? I think saying congratulations is so important, and we've got to get the money. The money means everything, and I was fortunate to have as the late mentor, the late Mary Lasker, who taught me that if you want money, you've got to go to it, and you've got to go to your congressman and your senator over and over and over and over. And we do, by the way, that's very important. And you've got such promise ahead. We've got to keep this work going, and anything I can do to help, count me in.
Awesome.
Thank you very much. That's really great to hear. And I got a bunch of really awesome students who are making it happen.
All right, then I'm going to turn it over to Erin to introduce the Women in Science panel. So if I can get all my panelists to turn on their cameras, we'll go ahead and introduce the session, and then everybody stay tuned because we have a lot exciting session to continue.
Okay. First of all, here's the award again, in case whoever wanted to see it again didn't see it. It's really hard to show because it's reflective. All right, and before we move into the Women in Science panel, the Genome Writers Guild would like to invite everyone to a weekend screening of Picture a Scientist. Picture a Scientist is a 90-minute movie that chronicles gender bias in science and provides new perspectives on how to make science itself more inclusive. And it also stars our very own panel member, Nancy Hopkins. Tickets should have been emailed to you today. If not, you can find the link on Canvas. You will have from after the panel tonight until midnight Sunday night to view the movie. So again, the link should be on Canvas if you can't find it in your email. And we'll start off the Women in Science panel with all of our panelists giving themselves a brief 2-minute self-introduction, which will be followed by the Q&A. Please put your questions in the question and answer section and include the name of the panelist whom you would like to address the question to. If you would like the questions to go around the whole panel, please type all in front of your question. And with that, Ms. Marianne, will you start us off?
And you're muted. Yep, there you go.
Okay. First of all, I'm just so excited about what we're doing tonight. And I wish that Rita Caldwell could be with us this evening because she is the president of the Rosalind Franklin Society. And as you know, she is so highly esteemed and as a marine biologist, and her new book, A Lab of One's Own, is coming out in August. So we're all really very excited. And I think it's important, I want to say that, when you look to the future, people will remember James Watson, but maybe for the wrong reason. Already, if you mention Francis Crick without Watson and Crick, people don't always remember. But we're going to make sure that Rosalind Franklin, who didn't get remembered for her contribution to the double helix, is remembered, and her name and her memory will continue to be celebrated because she made a difference, and we're going to make sure that that is perpetuated.
All right, and then we'll have Nancy go next and give us a few words about yourself and kind of where you are.
Okay, thank you very much. I wanted to thank the organizers for this terrific event. And I also wanted to say I thought that talk was fantastic. Thank you so much. Oh my God, I wish I could start all over again. I wish I was young, start being a scientist again. Anyway, it was just fantastic. I also wanted to thank Marianne because, yeah, I think that the Rosalind Franklin Society has really made a difference. And I think this idea that women should get recognition. When I started, there were actually already successful women scientists. We just didn't really know about it because they weren't recognized. So I think focusing on this issue of recognizing women for their contributions has been tremendously important. So thank you so much for doing that, Marianne. Really important. And so me, I was a molecular biologist, and believe it or not, I was inspired to become a scientist after hearing one lecture by James D. Watson. So there is a funny connection to the Rosalind Franklin Society. I actually read early editions of the Double Helix when I was a student, and he was writing the book about Rosalind Franklin. So I know a lot about it, about that topic. But I was a molecular biologist, and I worked in 3 different areas of science over a long period of time. I worked in phage, and then I worked on cancer viruses, and then I worked in zebrafish. And now I'm an emeritus professor from MIT. And along the way, I became, against my better judgment and wishes, an activist for women in science. And I did it because I literally couldn't do my work. I would not have been an activist, but I found there were so many obstacles to getting my research done that I had to become an activist. So I did, and ended up working with the other tenured women faculty in science at MIT in the long time ago, starting in the 90s. And we issued a report in 1999. And I thought when I first got interested in this that I was one of maybe 3 or 4 people in the universe who cared about this problem. And I was so naive. Oh my gosh. Anyway, we issued this report and discovered this was a universal issue. And so I became an activist and stayed with that issue up till today. Really, here we are again. And honestly, I thought by the time I became emeritus, this problem would be solved. But obviously, from the comments Alexis made earlier, I was shocked to discover the problem has not gone away completely. But it has improved enormously. So I'm really looking forward to hearing the comments of younger people tonight about this issue and where we are.
Thank you so much. Samantha, we're going to have a couple minutes from you.
Hi, I'm Samantha Maraj. I am by training a human geneticist and molecular biologist. I had maybe a little bit of a different path than many. So I did my undergraduate in biology, and then I got a job. So I actually started working as a technician with the federal government and at NIST, where I work now. And then got the opportunity to go do my PhD and then come back to NIST. And so I've worked both as a technician at a BS level. I got a master's while working, and then did my PhD and came back and have had the opportunity to start labs and programs at NIST. I've spent my entire career at the federal government, so that's the window and the view that I have. I will say that I didn't know from my training that there were opportunities besides academia. And so that's one of the things that I didn't have anybody tell me. People thought I was crazy when I said, well, maybe there are other things. So the concept of government or industry wasn't something that was really touted. So I sort of had to forge that way a bit more. And then now started the work that NIST does in genome editing. So starting sort of a new competency area at the organization from scratch, and those sorts of interesting challenges to break into, both a new area where you're trying to find new colleagues that you didn't know before, where your organization didn't actually have somebody that was doing that previous to you so that they could introduce you. So those sorts of interesting things when you're a woman in science, and then having to advocate for yourself to a new set of people, are some of the experiences that I've had. So I'll also mention that I've the issues of being a woman in science and also being a minority woman in science also poses some other interesting challenges and experiences as well.
Yeah, we're definitely going to be interested in hearing some of your viewpoints on that in the panel as well. I think we lost Tanya. She's not on the, I don't see her. Tanya, are you here? If you are, say something, but I don't see her.
Tanya's had a last minute emergency and has had to leave the call, so she won't be on the panel tonight.
Okay, hope everything's okay. All right, so then I'm gonna, Alexis, do you want to say anything? I think we introduced you, but I would be happy to hear from you too.
Sure. Yeah. I guess I'll just say, I grew up in Northern California and went to UC Berkeley for undergrad, and then where I was an inorganic chemist. And then when I went to grad school, I wanted to do bio-inorganic chemistry, and I only felt comfortable working for a woman. And so the only person whose lab I could join basically was Jackie Barton. And I'm really glad that I did, because she was an absolutely amazing mentor. If you look at her list of alumni, it's mostly women, and like lots and lots of female faculty members came from her lab. She's just an outstanding mentor and role model. And so after that, I kind of left the inorganic chemistry world behind, and then did a postdoc for David, where I said, oh, I want to learn how to do protein engineering and work with DNA binding proteins. And then he kind of tricked me and said, well, Cas9 is a DNA binding protein, so go work on that. So that's how I ended up in genome editing. But I like being here. It's awesome. I also, if judging by the list of finalists for this award, if that was sort of like all of the women in genome editing, like young faculty, that's really sad, because if there was an equivalent award for early career men in genome editing, it would be pages long. So yeah, I was with David's lab for I think 2.5 years, and then moved back to the motherland, to California, to start my independent career, and here I am.
And I will say, those were just the finalists, so there were lots of nominations. And so, but you guys, we picked, we wanted to recognize all the people that there were some really great ones, and we wanted to make sure that we gave them recognition they deserve as well. Yes, there could have been more, and there should be more. All right, then I'll move on to Ava.
Hi, everyone. First of all, I want to say congratulations to Alexis and the other finalists. You are an inspiration for all of us. In terms of my background, my name is Eva Galanis. I'm originally from Greece, where I did my medical degree and my DSc degree. Then I came to this country for additional training. I'm an oncologist. I'm also a researcher. I run an NIH-funded laboratory for the last more than 18 years. Our passion in the lab is engineering viruses, oncolytic viruses, and cells for cancer therapy. In terms of roles at the Mayo Clinic, I'm a professor of oncology. I'm leading the gene and virus therapy program of the Cancer Center. Also had the honor to be the chair for the Department of Molecular Medicine here, one of the basic research department for 10 years. And then I transitioned on to become the executive dean for development here. And in terms of, a role that I'm quite a bit engaged in outside Mayo is chairing the Brain Tumor Committee for one of the NCI-funded cooperative groups who have more than 600 sites. And what we are trying to do is basically take innovation and discoveries in brain tumor research and move it to the clinic and accelerate new drug development. So I have had the benefit of great mentors, and I'm also very passionate about mentoring others. So I'm really honored to be part of the panel tonight. One thing I want to remind us all is the etymology of the word mentor. So mentor in Homer's Odyssey was this wise advisor of the son of Odysseus, and was actually Athena, the goddess of wisdom, who was disguised as this old man to guide the young Telemachus to navigate this very difficult situation of having to deal with his mother being surrounded by all these prospective rulers of the small island of Ithaca. So if we look at this metaphor and what it means for all of us as women, as scientists, the path we are taking, and our role in inspiring the young generation, it's just such a privilege. And again, thank you for including me.
Thank you for being on the panel, for sure. I'm going to move over to Steve Ecker, and he is, as you can tell, a man. But we're very happy to have him as a woman advocate on the panel, and we also wanted to have a man on the panel just because it's really important to include them in the conversation. So we definitely need a perspective from him, and he has, even in the time I've known him, he's been a very, very big woman advocate. So I'm going to go ahead and let Steve introduce himself.
Well, thank you. And I want to say congratulations again to all of you. I've known Nancy a long time, and it's been awesome to have her as a scientist colleague, and then to help transmit her signal and amplify her signal. That's been important all of my scientific career with her. So it's been. She's of course does zebrafish work, so I have to say it. So I'm Steve Ecker. I am the founding president of the Genome Writers Guild. I am excited by this partnership with the Rosalind Franklin Society. I love this medal. I think it was the opportunity to have it on the 100th birthday, which is tomorrow, for Dr. Rosalind Franklin seemed like a perfect, wonderful, positive thing. I know we're doing this virtually. I would prefer to do this in person, but perhaps by being virtually, we're all able to meet and connect. So I think that there's positives. I have a couple of hats. I still do genome editing and genome engineering, but I also am the dean of the graduate school for the Mayo Clinic. And I really, our push is to develop leaders, and this is a part of our domain. It's leaders in biomedical sciences and education, and we need the leaders to represent the demographics of our community. And we know that that's just not true. All you have to do in academia is look at the professorial ranks, let alone the dean ranks. If you look at the CEOs, right, in Fortune 500 companies, I don't even need to, there's more Johns or something in Fortune 500 than there are all women, right? I mean, it's super sad, right? And I think of it always in a positive light. It's the opportunity to untap all this potential that's clearly not being tapped. Think about where we've gotten, but imagine where we would be if all of that potential had been untapped. And I think that it is selfish of me to sponsor and support this. And so I'm excited to do so, and I'm delighted to have this. And then I'm going to just be quiet. If you have a predictive question for me, otherwise my job is to be cheerleader and to make this and to make sure this happens. And I'm so excited for that. And I'm looking forward to watching the Picture Scientist. It's coming up.
Awesome, thanks so much. So we're gonna, for the session, for the rest of the questions, Erin and I are gonna go back and forth and ask you questions. I would encourage the audience, again, as Erin said, if you have questions, to put them in the Q&A. And you can put the specific person that you would like to ask the question to, put their name first. Otherwise, if you'd like the whole panel to go around, you can put all in that. So I'll go ahead and pass it over to Erin for the first question.
Okay, and I'm gonna open it up with a question to the whole panel, sans Steve. Ladies, what are some of the unique barriers that you have faced as a woman in science that your male colleagues likely aren't even aware exist?
And let's go ahead and start in the order that we went. So Marianne, if you can answer that first.
Okay. Would you repeat that, please?
Sure thing. Can you describe a unique barrier that you've faced as a woman in science that your male colleagues likely aren't aware even exist as a barrier for you?
Well, it's a hard question to ask, really, to answer, really. I'm very passionate about women in science, obviously. I'm passionate about science, and I'm worried about science. I think, we're not encouraging enough young people to go into science. I think it's hard for people in science to stay in science, and I worry a lot about the money. By the way, I want to mention that Ava, you should see if you could get a Vilcek Award. I think that you are very well qualified for a Vilcek Award, and you meet the criteria that they are looking for.
Thanks. All right, then we're going to ask the same question to Nancy. What are the barriers that you've faced that men haven't faced or don't even realize they exist?
Well, I think it's a great question, actually, and a very sad question. Okay, here's what I found out. I mean, when I started, when I was young, that's why I was very shocked by what Alexis said earlier, but when I was young, I did not think this problem existed. I had no concept there was such a problem, actually. I thought the only problem women in science faced was the family-work balance. And in my generation, it was so much more severe than it is now. I mean, you didn't even mention the word baby. It was not mentioned, didn't exist. I mean, so you solved that independently on your own, or you weren't a scientist, then forget it. So that's changed enormously, but I thought that was the only issue. So the problem that I've seen, and that took me so long to figure out myself, and therefore I'm sympathetic to other people who haven't figured it out, and men, and that is, it is this marginalization and undervaluation of women. And I think the issue is double for minorities. So I think it's a similar issue, but more extreme. And I remember when we were doing this work for women in science, I ultimately ended up working very closely with the president of MIT, Chuck Vest. And he established a committee, and I was appointed to the highest level of the university to work on this problem with him and with the provost, who was then Bob Brown, who's now the president of Boston University. And one day, Chuck, if he was in town, he would come to our meetings where we were discussing how to address the issues that women had. And there were all these technical issues that are the fallout of the problem of gender bias, such as space and resources and hiring and so on and so on, all these, the fallout from it. But the problem really underlies it is this marginalization and undervaluation of equal or even superior work by people. And it's a hard thing to fight, because how do you prove it? And it's almost impossible to fight. So you have to be better, doing better, working harder to achieve an equal amount. And Chuck one day said to me, Nancy, he listened to all of this, he read the report, he believed it, he understood it extraordinarily well. And he said, anything I can measure, I can fix, but I don't know how to fix the marginalization. And here we are 20 years later. And it's still true. So he set about it. So MIT took the issue on, and they did fix all these things. They put daycare on campus, on and on, family leave policies, hiring policies, the works. And it really worked. You increase the number of women, they're happy, they appointed them to this, that, the other. But it's your colleagues, it's the people you deal with on a daily basis who still marginalize and undervalue the marginalization people that they're working with. And we still don't know how to fix it. And I was shocked, really, I guess, when the National Academy of Science report came out about 2 years ago. And I thought, oh my gosh, I read this thing and I thought, is that it? We haven't made any progress at all. I mean, they made it sound as if we really had. That's not true. We've made tremendous progress, but we don't know how to fix this problem. And where we see it now at MIT and the biological sciences, and I've talked to Marianne about this, is in the biotechnology industry. It's absolutely astonishing. But if you ask, okay, at MIT, founding companies is a huge part of the biology department now. Faculty, it's a common activity, professional activity. And half the men in the department are founders of companies. So I don't know if you care to take a guess of how many companies have been founded by, our faculty is about 65, 70 people. And anyway, I'll tell you, collectively, this faculty has founded 56 biotechnology companies. So how many of those companies do you think were founded by women who comprise a quarter of the faculty? Out of 56 companies, Marianne, what's your guess? Well, the answer is 2. Okay. 56 companies, 54 founded by men, and 2 founded by women.
Yes, and it's about time. By the way, I think one of the barriers.
Did you unmute?
Pardon me?
Did you unmute?
Oh, did I unmute? Hold on.
Yeah,
She
you're
did.
muted.
She did.
Yeah.
She
I'm
unmuted.
unmuted, right?
Anyway, so I mean, the point is that, and many of these companies are founded by male faculty, founding companies with other faculty. And if you ask who are they founding with, we looked at several departments, there were 27 people, faculty involved in co-founding companies. Not a single woman faculty was included in those co-foundings. So 27 out of 27 were men. This is in the field of biology where, as we know, there's no pipeline problem. So those things just happen in the workplace to this day, and we do not know how to change that, in my opinion. So.
Well, Nancy, you know, I think one of the things is that women are afraid to ask for money. That's number one. And they are also somewhat concerned about being noted as too pushy. When Joe Handelsman was president of the Rosalind Franklin Society, she told me the story of when she wanted to be nominated for a prize. She went to the head of her department and asked him to nominate her, and he said, "I'm too busy now, ask me again next year." And the answer is, that is not acceptable. That is not acceptable. Women have to know how to ask, they have to insist. Another barrier, by the way, just to give you this one more barrier because it's important, is that when men go someplace, they find a job for the woman's spouse. When women are asked to take a position, they have a harder time sometimes finding something for the spouse if they're not in science.
That's a good point. Let's go ahead and go to the next question. I'm gonna start with Samantha for the next question. When have you had an experience where you had a male advocate that actually was advocating for you and that was a good experience? And did you learn any lessons from that? I know that's a very hard question to answer on the spot. I will preface it, or I'll also tell Steve, I'm gonna ask him to answer the question kind of the converse. When have you been a male advocate and that led to a positive outcome?
So I actually have a very clear story for this in my case. I mentioned in my intro that I initially came to NIST where I am now as a technician straight out of my undergrad with a BS. And I was working as a technician and I was doing that. And I was doing a master's at night, but in my organization, this would have been my boss's boss's boss. So he was the boss of the 1,000 of us. And one day there was an event that, had people mingling of different ranks, and he tapped me on the shoulder. I didn't really know that he knew that I existed. He tapped me on the shoulder and he said, "You should go do your PhD." And I said, "What? You know who I am?" And he had been somebody who was watching out for trying to foster, the next generation of young scientists in our organization. And I initially was like, I like my job. I like being here. And he pushed me and said, no, I'll support you. You should go do this and we'll make sure to support you. And he's been a fantastic support and advocate all the way through. He ended up advancing and becoming the head of the agency at one point. But that was somebody who, in my case, saw something, was looking to promote and support, and came to me and said, with an opportunity, and followed through on supporting all the way through, the process.
That's awesome. All right, Steve, you're up. When have you had an experience where you felt like you were able to advocate for a woman, and what were the lessons that you kind of learned?
It's a great question. I'm just going to put it this way. I've never regretted being an advocate, ever. It's always been positive. If you find talent and you can help them, they grow. And it's really easy. It's one of those easiest things in the world for me to spot and see. And but I'll give an example at my own institution. We brought in Dr. Lisa Scamenti. We hired her from the University of Minnesota a few years ago, and she's now department chair, really taking on clinical genetics in a very important way at Mayo. But that was the last search committee I was on. And the next, you want to take a stab at how many of the next hires were men?
All.
19. The next 19 hires were all men. I'm sorry, this is wrong, right? You have to have people on the search committees, and it has to be guys, not just women, advocating for top talent. How else to say this, right? This audience is there's a lot of, I've been looking, there's a lot of men in the audience, right? Everybody has to look around and scenario. It's never been regretted. I never regret this scenario. But you actually have to pause and say, okay, look at the whole person in front of you, right? Take a step back. Don't assume cultural scenarios. Don't assume these other pieces. Pause and reflect. And in science, if you've got a rubric, you can start focusing on the rubric. And the other things can become minor. So, but those are my views. I've never regretted it. I've always, I only regret not being able to do more, because it's so obvious when you take a step what the benefits are, and the benefits to all of us.
Aaron, do you have the next question? Oh, you're muted.
Okay, sorry. We have a question from the Q&A from Allison Van Enenam. Alexis, I am horrified to hear you say that sexual harassment continues to be a big problem for your generation. Nancy says things have improved. Have they? In your experience?
Well, I don't know how bad they were, back when Nancy was in my situation, but I mean, I'm happy to say, in my current position, I haven't experienced it at work. I have experienced it at conferences. There were, 2 instances at like different men, different conferences where I was like followed around. And like in one point I had to call my husband and tell him to show up to like get rid of this guy who just wouldn't leave me alone. And then another time I was a panelist, and this guy like sat next to me and was like flirting with me or something. And I kind of like very politely took out my laptop and said, oh, I have to work. And then he like took out his phone and was taking pictures of me for the rest of the panel while I was up there. And it was like I just felt super icky and left like as quickly as I could. Whereas, normally my male colleagues would spend that time to network and to not worry about these things and to just focus on the science and meeting new people and stuff. So it's, and think about every time I go outside to exercise, I go on a run around the neighborhood. I'm not thinking about science. I'm worried about if I'm gonna like get harassed in some way. So I think that's something that men don't really understand, that they have all this time in the world to be thinking about their science. Whereas if in my case, I'm worried about keeping myself safe. And it's like definitely something that I have to deal with. I imagine it probably was worse in previous years. I mean, I don't know, but it's still definitely really bad.
Can I add something to this?
Please.
I have not had a pervasive issue in the workplace, but I have definitely had the issue in the workplace. And in my situation, it's kind of sad because it's sort of, what's that line? You're like, well, you create some line to say, well, they haven't done this, and you keep making excuses because you're not really sure what to do. And in my case, it was somebody that was in authority to me. And so there's that challenge as well as to, well, what will this mean? And, will they believe me? And that sort of thing. And this was in a science context with, in the workplace. And I agree with what Alexis said, is that I'm constantly thinking, particularly when I'm on travel, about these sorts of safety things. And, other people might feel comfortable, networking or going out after this and this. And I like, well, we'll have to walk back to my hotel by myself. And what does that look like? And, who's around? So it is constantly on one's mind as a woman that male colleagues I don't think about these sorts of things. And the more that I've had conversations, the more I find other younger women in science have experienced harassment, and then we don't know because you don't say it. And it's not that it's not happening, it's that, well, who would I say it to? Why would I say it? What will people think of me? And will that be what they remember about me? And so yeah, I from my experience and speaking with others, I think it's more of a silent thing that doesn't really get addressed because people don't speak up.
I think that's a really good point. And I think Nancy, in this movie, I think Nancy has some things that she says in the movie that I was very shocked by. And I think I really applaud you guys for bringing it out in the open, because if you keep it in the dark, it will always be in the dark, and bringing it to light is really important. So thank you guys for that. I'm going to ask Ava a question. Marianne mentioned that we don't, as women, oftentimes we don't advocate for ourselves and we don't ask for things. Ava, have you had an experience where you looked back and thought, oh, I should have asked for more? Or have you felt yourself negotiating something and feel like you were intimidated to ask for what you thought you might really deserve?
I think that's part of the process of growing up, right? And then you pleasantly discover then when you're not afraid to come forward with your thoughts and ideas, people appreciate that. I think there are a couple of issues related to that. One is not a lot of role models. I have had a number of mentors, of them only one is a woman, and no mentor in science who was or is a woman. And I think that's critical. Having the being surrounded by women who have taken the path before you, like the panelists today, I think that's critical. That will give our young scientists their voice, because they're seeing other women who have taken this path, who can speak up. So and also I think it's a reminder for all of us. I feel that sometimes as women try to advance in science, they're undermined not by their male colleagues, but sometimes by other women. So that the bar sometimes can be higher. The bar we raise for our colleagues. And I think, again, that's an opportunity there to serve as mentors. As I say sometimes to my mentees, free up your mind. The possibilities are endless. You just need to feel secure enough to ask for it.
Okay, we have a question from the chat here. It's not addressed to anyone in particular. What can we do to showcase more of the outstanding women in science? They are there, many superstars, but how can we make this more obvious? Marianne, we haven't heard from you in a bit. Would you like to answer? You are muted. You are still muted.
I think there she is.
All right.
We can hear you.
There was something called Rock Stars of Science. I don't know how many of you remember that, but it was held in Washington, D.C. It was sponsored by Research America, of which I was a founding member, but I wasn't a member at that time. And none of the rock stars of science were women. And so I called the sponsor, because it was a corporate sponsor, and I said, no. If you're talking about rock stars of women, rock stars of science, women deserve to be on that too. And they were after that. And I think that we have to make it known that women must be appreciated, they must be promoted, they must be mentored, and they must speak for themselves. And if they see something like Rock Stars of Science in Washington, you can't just sit still. You have to be part of the movement that ensures that women will be equally recognized and valued. And that's not easy. That's not easy, but it's an obligation. It's an obligation to yourselves and for the scientists who are going to follow you, and as a tribute to those like Rosalind Franklin who've come before you.
All right. What about, I wanted to kind of talk about babies, having kids. So I don't know how many of the women on the panel have children, but if you do have children and you feel comfortable speaking about it, like how did you manage being such successful women and having children? I'm gonna have to let you guys jump in because I don't know who has kids. I have two that are running around in the background right now, so sorry if you hear them.
And I mean, I can speak. I have one child, one daughter. It takes a village, I think, I mean, as Nancy said, for the greatest part of my career, I thought, you know, one of the greatest problems was how do you balance everything? So at least, and I think there are a lot of strategies, and I'm sure that people who have been able to succeed in science, they have found ways to navigate that. But at least for me was to get as much help as I could for things that I could hire someone to do. So I could focus on my family and my work. But it's extremely difficult. And there is no such thing as being able to escape guilt. As a woman. I think my husband is managing that much better.
Can I?
Marianne, you are now muted.
Oh, no, okay. My husband is a pediatric surgeon. And I have noticed over all these years, when he would get a call in the middle of the night, or when somebody takes the child to the doctor, it is the woman. And when it's a call from the school, it's generally to the woman. That's the way it is. Now, is that changing slightly? Slightly. Is it changing enough? Maybe not. But it's harder for women who are trying to have children, to have a career where, as Matilda Krim said, the cell waits for no one. In fact, Claire Fraser-Leggett, at our first, who was married to Craig Venter, and she was at our first meeting of the Rosalind Franklin Society, and she said, I was working so hard that I had to make a choice. And so I chose my husband's career and company. She said, I hope this is a choice that I will not end up regretting. So I've never forgotten that. And I think it's so important to try to ensure, and I don't know how to do it, but we have to do a better job of making sure that childcare does not interfere with career advancement. And it's very difficult. As a matter of fact, I think it's probably one of the most difficult if you want to be a parent and a scientist. Of course, today, maybe hours are different. It's not 24/7 anymore, but in some cases, it really is 24/7.
Nancy, you had something you wanted to say?
I did want to say this is one of the things that I really saw change. There were several things that really changed in my lifetime, and one of them was this. I mean, because as I say, I thought it would not be possible to have children and do the job I saw these men doing. I worked in Jim Watson's lab when I was an undergraduate. I looked around, I was there a week, I looked around, I saw these men were working 7 days a week. They had wives already at home taking care of children. How could you possibly do both? It was obviously not possible. And I assumed that was the reason there weren't any women on the faculty. And then when I became a scientist and joined the faculty at MIT, I made a conscious choice not to have children, actually. And there were so few women, and more than half of them did not have children. And at that time, it was believed that you could not be a great scientist and have children. It was just not possible because there weren't enough hours in the day. So women actually hid their pregnancies. I mean, that was where we started. And one of the major things that happened as a result of this work we did in the 90s was that discuss this issue, bring it out on the table, and we could actually talk about it. So today, most young women who come to MIT, they interview, they're often pregnant when they interview, which I couldn't even imagine. The first time I saw it, I said, oh my God, that's incredible. And we put daycare on campus, the babies on campus. I pushing a man pushing a baby carriage on the campus of MIT. I can't tell you, I was in tears. Anyway, so now the junior women, they almost all of them have children. So something major happened. That made it possible. And I think having the daycare on the campus, having family, no woman when we started in the 90s had ever taken a family leave and gotten tenure at MIT. Men took family leave and used it to start more companies or do more work. Women didn't take it because of the stigma attached to it. So there's been a huge change. Women do have children, they take family leave, they ask for things, and that's tremendous. But it's still too hard. And there's still a difference, as Marianne was pointing out. So the next generation has got to keep pushing to get whatever it is they need that makes it more possible for more people to really do this. And I think obviously men are much more equal partners now than they were in my generation. So those things have made a huge difference. So it is really possible, but too hard.
Well, you know, Nancy, I think it also depends. MIT and places like MIT and UC San Diego and Princeton, these places have resources, but a lot of smaller universities and colleges do not have the resources or the budgets to help women with childcare. And so it makes it very difficult for them if they're not applying to one of these major institutions where women have more opportunities than practically any other place. It's not the same all over.
Okay, I'm gonna ask another question from the chat here. It seems like one of my limitations as an early investigator / physician scientist woman is the fact that I can't help caring more about the well-being of my communities, home, lab, department, institution, neighborhood, etc. And my need to devote time to these activities, which ultimately will not contribute to my promotion. The advice I keep getting is to say no and to not get too involved and to focus on papers and grants. I see it as a good thing that I'd prefer my institution recognize the time I put into my communities. Any advice? Samantha and Alexis, we haven't heard from you in a minute. Would either one of you like to jump in?
I don't have any advice. We have a word for it, at least. It's called invisible service. And it encompasses that as well as, you know, all of these sort of responsibilities that you have for, you know, being of the minority, trying to be a role model and trying to, you know, take your job as a mentor really seriously. And so sometimes some places I heard they're doing, you can have, you can submit with your tenure file, like letters from your students, say like, you know, praising your mentoring ability. Sometimes schools ask for a separate document where you describe what your mentoring activities have been. I mean, I think that, you know, we want people who take the mentoring aspect of the job seriously to have these higher up positions. And so I think, you know, having universities and you know, institutions to industry recognizing that and having it be you know, one of these things that they take into consideration when promoting people, that would be a step in the right direction. But I know it's really difficult to quantify that. It's probably easy to lie about it too. So I don't have a good answer for you, but that's my opinion. You just have to do the right thing, be a good mentor, and try to, you know, get the next generations of scientists excited about you know.
Did anyone else want to answer the question, or should we move on to the next question?
I just want to say that I think following your passion is energizing, and sometimes there are more than one ways to reach your target. So if you're passionate about making a difference in the community, in I would say go for it. And sometimes you find that would lead to other paths that sometimes you never imagine. I think the problem is when you do things artificially, or someone does things artificially because they have to check the box. I found that kind of sometimes being counterproductive. But what you're just mentioning, I think you have to trust yourself that you will find a way. That's how I would approach it.
Okay, I'm going to ask the next question to Samantha, and it's from our Q&A. We have a lot of questions coming in. So Samantha, do you ever feel like you are spoken to differently in the professional setting because you are a woman? And I'll even throw in because you're a minority.
So the reason gets difficult to deconvolute, but I don't know if it's spoken to differently or in body language and in sort of who you're gearing the direction to, is that you're not the one being spoken to, even though you're there. And so sometimes when there's a mixture in the room, it's like no one's looking at you. They're looking at, a male colleague, and they're directing the conversation at them. And sometimes even when you speak up, you sort of still don't get the attention in that moment. It's still not really, you're not really considered an equal partner in the conversation. I've experienced that at times. But I can't say spoken to differently. It's more so whether I'm considered, I feel equal in the conversation. I think that, and I see that most in body language, not necessarily tone of voice.
We have two questions in the chat that kind of go well together, so I'll ask them as one. Do you have any specific tips on how to overcome the feeling of being too pushy when you are simply advocating for yourself? If so, how do you address this? And tied with that, what actions do your mentors take, male or female, that built your confidence and initiative, built your confidence in taking the initiative the most?
So I have a quick response to this. I had one of my mentors from my organization. She had the opportunity to give a talk and speak on a panel. And one of the things that she said was, even when she got to, pretty high up in the organization, she still hadn't realized and learned that she was supposed to negotiate when she got that next promotion and to self-advocate. And it was really helpful to hear from somebody that I looked up to as a mentor, that they had this challenge and this was something that they were dealing with, and to call it out, to say, you have to do this, and you're not looked at differently because your male colleagues are probably doing this already, and there's not that mental barrier for them to ask for more or to speak up or to counter offer, because that's actually a normal part of the process to ask. And if you don't ask, you're not going to get it, because nobody knows. And my recommendation is the concept that, how will somebody know that you need that resource unless you ask them for it? They'll assume that you can do your good science and do what you need to do with what you have, because you haven't told them that you need it. And so I just sort of look at it that way. How are they supposed to know unless I ask for it? And they can tell me no, and that's okay. Sometimes you have to accept that no is going to be the answer, and you still have to be willing to ask again. You can't let no be the way that you get defined. You have to be willing to ask again and ask again. I'll also have to say, I'll put the caveat out there that I am very much an extrovert. I don't know how challenging that is for somebody who's naturally introverted. So I have more of an inclination to be okay, going up to somebody and maybe asking.
Would anyone on the panel like to self-identify as an introvert and speak up?
I'm totally an introvert, but I just want to add on to that. Like, the next time you do ask for something, don't apologize for asking for it. And someone pointed this out to me, and then I realized every single time I would send an email to someone saying that I needed something, it would always start with, "Oh, sorry to bother you," or, "Oh, like, I'm sorry that I'm taking up your time." So don't ever apologize for asking for something that you need. Just like if you screw up in a talk, don't apologize for it. Just say, "Oh, excuse me," or something.
That's good advice. This question I'm going to give to Steve. So as a man in a leadership position, you've heard some of these things. I know it's not the first time you've heard of this, but going specifically to what Alexis was, or the question that was addressed to Alexis, and that she was talking about community and, you know, this invisible service. What do you think you can do, or what can be done at the leadership level to basically get credit for those kinds of things?
So it's a really important question. I remember I was at the University of Minnesota, and which is a very proactive on paper organization for women in science. But I was on the panel and we did the analysis that in the first 10 years, the opportunity to delay your tenure clock by a year was baked into the system. And the only people that had taken advantage of it were men. And the women felt that it was not just stigma, but the reality was that the clock didn't really stop. So even though it was supposedly on pause for tenure, it might have been for tenure, but it wasn't for any other aspect of what they were on. So they were still expected to produce everything at the same level as the men. So there was no, it was completely artificial. So one thing I think we just have to reset. When people ask me about this, it's you value what you value. Okay, so if this is not a value to your organization, it's going to be a problem for you. It doesn't matter. It doesn't matter where you're at. Okay, and there's two things you can do, and one of the reasons that I'm here is that you can change your organization so that it is a value. So you can either change yourself or you can change the organization. It's hard, right? It's a hard question to ask. Are you changing the environment, like the soil, or are you changing the seed? And there's a really, Dr. Pam York is in Minnesota, and she's a big fan of Capita 3, and she helped bring me along this line.
Since we are not looking at them, again, I emphasize this, we're saying that the exemptions are very conservative in nature.
To change for the women to be successful. So it needs to be both, right? We need to have really outstanding women and diversity, right? Getting the experience, getting the training, getting the competency, these life skills that are not just how to write a paper, right? But we also need to make the environment around them successful, right? So if I'll just say, when somebody asked me this question, there was is it in your tenure book, right? I mean, it's really important that you make the tenure code, right? Because otherwise you're lost to the pipeline, right? But at the same time, you do need to follow your passion, right? And the best scenario is when you can make that overlap, right? That's what you're looking for. And so I look for the opportunities to help with the environment while I look for the opportunities to help with myself, right? And I think that's a skill set. That's a professional development skill set that we want everybody to have, right? And the men tend to get it, right? This is baked into their system. We understand, right? This is taught, this is part of leadership, right? But we don't teach this to a lot of women. It's not a natural, it's not in that, and then you lose, as you mentioned, right? The time, right? The how many faculty meetings are still at 7 a.m., Eva? I mean, I'm sorry, at Mayo. This is just ridiculous. There should never be a faculty department at 7 a.m. And so these things, these costs, these are taxes on the women, right? And there are missed opportunities, there are opportunity costs that were lost, all these things. So these are the pieces that you've got to do. So you've got to speak up for these things, and you've got to make them transparent. And I'm sometimes I feel like my job is to make it obvious to other people, like the 7 a.m. faculty meeting, right? This is just if you're a family, right, woman with family, what are you doing at 7 a.m., right? You're getting a kid on the school bus, right? That's what you're doing. And you know, I am, you know, I was lucky enough that my wife and I had a deal that I got the kids to the school bus, but she got them home. It's like, that's way better. That's the best deal I ever made. But it was okay. But I never went to a 7 a.m. faculty meeting unless it was really super duper, and you know, it was fine. But you know, but I think these are these kinds of things. I'd rather just not have a 7 a.m. faculty meeting. It seems like a wrong scenario. So it's a little bit long. There was a question around the impact of COVID on women's careers, because it's absolutely going to be more severe. Absolutely more severe.
I'd like to ask the panel in general for advice to young women who maybe aren't in science yet, and maybe they're in high school or middle school and don't very much have a good idea where they want to go with their lives. What advice would you give them if they're thinking about a career in science?
Let's have Nancy go. We'll have Nancy, you're going to wrap it up and then I'll finish the session. No pressure.
Well, I have to say that, you know, I debate this a lot now that I'm old and look back on my life, and I did find it hard dealing with these issues as they were much more, even worse in the beginning. But I have to say that if you love science, you just don't have a choice. I don't think I could have been a happy person if I hadn't been a scientist. I think science is, I just listened to Alexis talk tonight. I mean, I can get so excited about, it carries me away. It transports me. I'm passionate about it. And I think that science is the most exciting and wonderful occupation that there is. And I still feel that way about it. So if you love it, you just have to do it and just go for it. Don't even hesitate. And, you know, so many of the great jobs are in science today, and there's so many ways to be a scientist and so many types of jobs. We've really been talking about a very, very high, far end of the field, but there's many ways to be a scientist. So just do it, absolutely. And look for good mentors to help you along. And if you're lucky and you find good mentors, it's worth it. It's worth fighting for it, because it's a great life, truly.
You know,
What?
James Wood used to say, introduce kids as early as possible. He had grandchildren who were 5 years old, and he would show them science in a way that they can understand it. So it's important to make science exciting early on. Years ago, it was considered that men were better than women in math, or that boys were better than girls in math. In my case, that was true, so I went a different path. But I think it's very important to start introducing people to science as early as possible. They don't have to have a course in it necessarily, but when you show them something, you can introduce them to it. And I don't think we're doing enough of that. I don't think the schools are doing enough of it. And I think as individuals, we have to do more to point it out. For instance, years ago, the New York Times had a science section, but most people couldn't read it. It was too complicated for people to read. So we have to do it in such a way that people don't feel that they can't understand it. They're going to want to understand it, and they must understand it. So you make it simple for them, and then hopefully they will get more interested in it as they grow up, and can get their parents to encourage them too. Right now, parents encourage their kids to go into technology because that's where the money is. And, you know, that's where the money is. And the schools are reflecting that. So we've got to push for science. By the way, we have something called Go Green Shoelaces for Science. And the reason I put them out is we give them to everybody who wears sneakers. And everybody says, well, what are you doing with lime green shoelaces? And we say, those are Go Green Shoelaces for Science. And anybody who wants them, if we can ever get back into our offices, we're here to send them to you.
That's awesome. I just want to thank all the ladies and Steve for joining us on this panel. It has been really educational and sad and sometimes, and glimmers of hope for other things. I want to remind everybody in the session to take the poll. We're going to launch it just in a second. And please do take advantage of watching this Picture of Scientists film. I think it will be really, really good. We did, the Genome Writers Guild did pay a fee so that everybody who registered can watch it at no cost to them. So look for that in your email, and then it'll also be on Canvas. Thanks. Have a good night.